Welcome to Biology by Jamil Goraya. In Biology ATP, there are two kinds of graphs, line graphs and bar charts. There are certain steps in graph making: selection of axes, then label the axis, then linear scale on both axes, plotting the data, and the final step, joining all the plots.
The first step in making a line graph is the selection of the axis. This table is given, the data is given, the leftmost column, it always shows independent variable. And this rightmost column, it shows dependent variable. So we will always plot independent variable on x-axis and dependent variable on y-axis. So independent variable on x-axis and the dependent variable on y-axis. Now, after selection, we have to label the axis. And labeling must be complete like the given statement. On x-axis, we will label glucose concentration percentage. So let's start. Glucose concentration percentage. Similarly, the data given on y-axis is calorimeter reading / arbitrary units. So we will label the y-axis same like given. Calorimeter reading / arbitrary units. This labeling will give us 1 mark. Next step is a linear scale. Linear scale means on x and y both axes, the scale must start with 0 and it must have equal gaps. So if we see x-axis, it is from 0 to 2.0. So let's see how we have to make a linear scale. 0.0 like it is given in the table, and at equal gap 0.5, then it is 1.0, then it is 1.5, the next is 2.0. This is a linear scale with equal gaps as given. On the y-axis, if we see the readings, they are starting from 0.00 to 0.95. So we will be making a linear scale. It must always start with 0, separate 0 for the y-axis. So it is 0.00. Then it will be 0.20, then 0.40, then 0.60, then 0.80, and the last will be 1.00. So this is how we make a linear scale on x-axis and on y-axis. This will give a second mark. First for the labeling the axis, both x and y, and second mark for making a linear scale. Now third step is to plot the data. For plotting the data, what we do, we put dots as per the readings given. If we see the data at 0, the reading is 0.95. So what we will do is at 0%, reading is 0.95. This is 0.9 and this is 1. So in between will be 0.95. We will put a dot. Similarly, the next reading is 0.71 at 0.5. 0.71. This is 0.7 and above it is 0.71. Moving to the next, at 1, reading is 0.48. At 1.48, this is zero point 0.5, so below it will be 0.48. The fourth reading is at 1.5, 0.24. At 1.5, 0.24. Here it is 0.24. And the last plotting is at 2, 0.00. So at 2%, it is 0.00 dot here. After these 5 plots correctly, we will get the third mark. Now the last mark is for joining all the dots with ruled straight lines. Like this, then this dot to the next one, then to the next, and then the last one. So this will give us the fourth mark. This is how in line graph we have to complete 4 steps. Number 1, labeling the axis completely. Number 2, linear scale on the x and y axis that starts from the 0 with equal gaps. Number 3, plotting the data, clear dots at each, you can say, reading, and then joining all the points with a ruled straight line. This will give us 4 marks out of 4. This is an example of a line graph for the results of osmosis experiments. So let's see how we have to make graph of this data. First step is same, identifying the independent variable. The leftmost column, it is showing independent variable, percentage concentration of salt solution. So for your understanding, I'm writing it here, independent variable. And this change in mass, the rightmost column, this is dependent variable. As we know, independent variable is always plotted on x-axis, and the dependent variable will be plotted on y-axis. So the next step is, for the first mark in graph making, labeling the axis. And labeling is done with the complete statement given. Percentage concentration of salt solution will be on the x-axis. It's already labeled. Percentage concentration of salt solution on x-axis is labeled by the examiner. So on the y-axis, we will be labeling change in mass / gram. Here we will be labeling change in mass / gram. So this labeling will give us 1 mark. The next mark is for the linear scale. And if we see the data in table, for independent variable, it is from 0 to 8 with regular intervals. Same, we have to make a linear scale here on x-axis. So let's start with 0, then at equal gap 2, the next must be 4, next one is 6, and the last reading on x-axis is 8. So this is how we make a linear scale on x-axis. If we see readings on the y-axis, the dependent variable, they are in negative and positive. So we have to make a scale of both. So in positive, it is till 0.9. In negative, it is till 0.8. So let's see how to make this. 0 is already given. So above 0 will be positive values. So it is positive 0.2. Here it will be positive 0.4. Next will be positive 0.6. The next one will be positive 0.8, and here it will be positive 1.0. Similarly, below 0, we will make a linear scale of negative values. For negative values, same like the positive values, we will start with negative 0.2, then at equal gap negative 0.4, then with equal gap negative 0.6, then negative 0.8, and the last will be negative 1.0. So this is how we make a linear scale for osmosis graph. It will give us second mark. Third mark is for plotting the data. First, we will plot the positive values, then we will plot the negative values. So let's see the values. At 0, it is positive 0.9. At 0, it is positive 0.9. So here it will be 0, positive 0.9. Then at 2, it is positive 0.1. At 2, positive 0.1. So here it will be the second plotting. The third value is at 4, it is minus 0.5, negative 0.5. So at 4, it is negative 0.5. So it will be plotted here. The second last plotting is at 6, minus or negative 0.7. At 6, it is negative 0.7. So it will be here. And then at 8, I think it is negative 0.8. So we will be plotting the last one at 8, negative 0.8. So it will be like this. Plotting both the positive and negative values will give us third and fourth mark. And the last mark is joining all the points with ruled lines. So let's complete this with final step. We will be joining like this straight line, then this dot to the next one with straight line, then this to the next straight line, and the last straight line. So this will give us 5 out of 5. First mark was for the labeling the axis. Second is for the linear scale on x and y axis. Third is plotting the positive point. Fourth is plotting the negative points. And fifth mark is for joining all the dots with others with ruled lines. In this line graph, we will be learning how to make a line graph of two different data. Rules are same. First of all, identify independent variable that is always on the leftmost column. So this is independent variable, and the rightmost column, seed germinated, these are indicating dependent variable. The independent variable is always plotted on x-axis, and the dependent variable is always plotted on y-axis. Once we have selected the axis, next step is to get the first mark by labeling the axis. So as we see on x-axis, it must be time slash hours. So here we have to label time slash hours. And on y-axis, if we see seeds germinated slash mean number per petri dish, we will write the same. So on y-axis, seeds germinated mean number per petri dish. Exact labeling is needed to get full marks. So by labeling the axis, we will get the first mark. Now moving to next is linear scale, means starting with 0 and with equal gap. On x-axis, it is already with equal gaps of 24 hours, 0 to 96. So like here we will plot 0. Here it must be 24. And here could be 48, then 72, and here it must be 96. So this is linear scale with equal gaps. On the y-axis, as we have to plot both data, light and dark, so carefully look at the values. Both are starting from 0. It's maximum till 15, and it is 0 to 17. So very easy. If we start from here, 0, then with equal gap, here could be 2, here it could be 4, here it could be 6, the next is 8, next one is 10, next one must be 12, next will be 14, next will be 16, and the last one will be 18. So from 0 till 18 with equal gaps, this is linear scale. This will give us second mark, first for labeling the axis, second for linear scale. Now let's move to the third point. Next step is plotting the data. First we will plot the data for light and then for dark, and we will make two separate lines and label them. So let's start with this one, 0, 0, then 11. First value is 0, so we will put a dot at 0. Next one is also 0. Then I think it is 11. So it will be like here. Let's check the third value. 0, 0, 11, 13, and 15. At 72, it is 13. It will be like this. and the last value is 15. At 96, it will be here. Okay. For the dark one, we will be using a cross to plot the data so we can differentiate both. Let's see the values for dark: 0, 5, 15, 16, 17. Okay. So first one is same. So we will be putting at same place. Then is 5. It will be here. Then I think it is 15, 16, 17. So at 48, it will be 15 here. Then it is 16. It will be here. And last one is 17. That will be here. So by plotting these, we will get next 2 marks. And the last step is joining all the dots or all the crosses with each other by straight line. So let's complete it. First, I will be joining all the dots with straight line with ruler like this. Then this. This is for the light one. And just for your understanding, I'm changing the color. In paper, you need not change color. Just use same pencil and scale. The first one, the second one, the third one, and the fourth one. So this is how double data line graph are made, and how we can label them. It's very simple. You can write here, it's for the light, and this is for the dark. So this is how we will be getting all 5 marks in this graph. Third type of graph is a bar chart. Rules are same. First of all, identify the independent variable that is on the leftmost column, plant species as per given data. So this is independent variable. And this rightmost column, it is showing dependent variable. Now, independent variable is always plotted on x-axis and dependent variable on y-axis. Always remember this key point. Next step is labeling the axis. So now let's move to the graph. On x-axis, we will write plant species as per given data. And on y-axis, we will write estimated number of plants in the whole field. Exactly what is given. So on y-axis, we must write estimated number of plants in the whole field. So by labeling, we will be getting 1 mark. Next mark is making a linear scale. In case of bar chart, linear scale is only on y-axis. So you can see values starting from 3200 to 7200. But our linear scale must start with 0 with equal gaps covering all the values and covering more than half of the grid given. So it will be like this. Like if we start with 0, after regular interval, it will be 2000. Then the next must be 4000. The next regular interval must be 6000. And the next must be 8000. And next could be 10,000. This will cover all the values with equal gaps. Now, for the first bar, look at the data. It is plant species C and value is 5600. So we just need to write C here. And for C, we will be making a bar. $5,600. This is 5,000, 52, 54, 56. So the bar must be like this. Or if we make a bit wider one, then we will shade it with the pencil. Now we have to give equal gap and make the next bar for D species of the plant. So let's see how it works. Equal gap, then here it will be the bar of plant D. So if we see the values for D, it is 3200. So 2000, 3000, and 3200. So this bar will be till the height of 3200. The next one is for plant species E and value is 7200. So for E, we have to make a bar till 7200. This is 7000 and this is 7200. With pencil and scale, you will be drawing these bars and labeling as well. And this is E. So now you can see there are total 4 marks. First mark is for labeling the axis. Second mark is for linear scale starting from 0 and with equal gaps covering all the values in the given data. Third mark is for making the bars of equal width, neat, clean, pencil with scale. And then fourth mark is giving equal gaps between the bars. So this is how we get full marks in bar chart.